Next is chapter 13, Effector Mechanisms of Humoral Immunity. It will be a little bit long topicy, but interesting with some things that tells you how the immune system works, especially the humoral immune system. So, overview, we'll discuss the overview of humoral immunity. We will see the neutralization of microbes and microbial toxin, antibody-mediated opsonization and phagocytosis. We'll see about leukocyte Fc receptor, antibody-dependent cell-mediated cytotoxicity, antibody-mediated clearance of Helminth. We'll see complement system. That will take us some time to really finish it, because there are pathway for complementary activation, receptor of complementary proteins, regulator of complementary activation, function of complements and complements deficiencies, pathological effects of complementary system, evasion of complementary by microbes. All of these are the important things that we're going to learn. Apart from that, we're going to see the summary. So let us start. Humoral immunity is mediated by secretory antibodies, and its physiological function is defense against extracellular microbes and microbial toxins. This type of immunity contrasts with cell-mediated immunity, the other effector arms of the adaptive immune system, which is mediated by T lymphocyte and function to eradicate microbes that infect and live inside host cells. Humoral immunity is the form of adaptive immunity that can be transferred from immunized to naive individual with serum that contain antibodies. The types of microorganisms that are combated by humoral immunity are extracellular bacteria, fungus, and even obligate intracellular microbes such as viruses, which are targets of the antibodies before they infect cells or when they are released from infected cells. Defect in antibody productions results in increased susceptibility to infection with the many microbes, including bacteria, fungi, and viruses. Currently used vaccine-induced protection primarily by stimulating the production of antibodies. Apart from the crucial protective roles, antibodies can be harmful and mediate tissue injury in allergic individuals, in certain autoimmune diseases, in the blood transfusion reaction, and in the transplant rejection. In this chapter, we will discuss the effector mechanism that are used by antibodies to eliminate microbes and their toxins. The structure of antibody is described in chapter 5 and the process of antibody production in chapter 12. So, overview of humoral immunity. Before we discuss mechanism by which antibody provide a protection against microbes, we will summarize some of the silent features of antibody-mediated defense, antibody-mediated host defense. Antibody-mediated elimination of antigen involve number of effector mechanisms and requires the participation of various cells and secretory proteins of the immune system, including phagocytes and complementary proteins. Antibodies are produced by plasma cell in secondary peripheral lymphoid organs, inflamed tissues, and bone marrow, and antibodies can perform their effector function at site distance from their production. Antibody produced in the lymph nodes, spleen, and bone marrow may enter the blood and then circulate throughout the body. In mucosa organs such as the intestine and airways, antibodies are produced in the lamina propria, cell-rich connective tissue space that separates the innermost layer of epithelial cell from the underlying concentric smooth muscle layer, and transported across epithelial into a lumen, whereas these secreted antibodies block the entry of ingested and inhaled microbes. antibody are also actively transported across the placenta into circulation of developing fetus. In disease state, antibody may be produced in peripheral non-lymphoid tissue at the site of infection or chronic inflammation that are sometimes called tertiary lymphoid organs. The antibodies that mediate protection immunity may be derived from short-lived or long-lived antibody-producing plasma cell. Long-lived plasma cells reside mainly in bone marrow. In cell-mediated immunity, activated T lymphocytes are able to migrate to peripheral sites of infection and inflammation, but they are not transported into mucosal secretion or across the placenta. Therefore, antibodies are the only defense mechanism used to combat microbes in lumen of mucosal organs and in fetus and newborn. So vaccine-induced immunity, this is the table that is just explained. So vaccinated induced immunity, we see there is an infectious disease, vaccine, mechanism, protective immunity. So we have the polio. This is injected inactivated polio vaccine, which is called Salk, and oral attenuated polio vaccine, Sabin. So mechanism of protective immunity, neutralization of virus by IgG or by mucosa IgA antibody. The tetanus and diphtheria, these are toxoid inactivated toxin. The main mechanism is neutralization of toxin by systemic IgG antibody. Hepatitis B and A, they work recombinant viral envelope proteins. They produce antibody for neutralization of virus by mucosa IgA or system IgG antibody. Pneumococcal pneumonia, this is a conjugate vaccine composed of this is the conjugate vaccine, and apart from that, pneumococcal pneumonia, there are Haemophilus influenza, the bacterial meningitis caused by Neisseria meningitis. All these ones, they are conjugate vaccine composed of bacteria capsule polysaccharides attached to a carrier proteins. So what they do is they do opsonization and phagocytosis mediated by IgM and IgD antibodies directed secondary to complement activation. So this is just example, selected example of vaccine that works by stimulating protective humoral immunity that's they have been listed. Many of the effector functions of the antibody are mediated by Fc regions of immunoglobulin Ig molecule, and that and different Ig heavy chains are observed to distinct effector function. For instance, some IgG subclasses IgG1 and IgG3 bind to phagocyte Fc receptor and promote phagocytosis of antibody-coated particle IgM and some subclass IgG. IgG1, IgG2 to a limited extent, and IgG3 but not IgG4 activate the complement system, and IgE bind to Fc receptor of the mast cell and trigger the activation. Each of these effector mechanisms will be discussed later in this chapter. The humoral immune system is specialized in such a way that different microbes or antigen exposure stimulate B cell switching to Ig isotope that are best suited for combating these microbes. The major stimuli for isotope switching during the process of B cell activation are cytokine together with the CD40 ligands expressed by activated helper T cells. Neutralization is the only function of antibody that is mediated entirely by binding of antigen and does not require participation of Ig constant region. So this is just a diagram. Neutralization of microbes and toxins, opsonization and phagocytosis can happen. Then Fc receptor natural killer cells can kill. So if antibodies have, if B cells, they have produced antibodies and microbes have been attached to B cells and coated with antibodies, it can be phagocytes with can be killed by natural killer cells. Anything can happen, and there are many long way to go. So effector function of antibodies, effector function of antibodies against microbes and the toxin not shown neutralize these agents, opsonize them for phagocytosis, sensitize them for antibody-dependent cellular cytotoxics, and activate the complement system. This virus effector function may be mediated by different antibody isotopes. Natural killer cell also is involved. So function of antibody isotopes, we have antibody isotopes effector function. IgG, this is for opsonization of antigen, for phagocytosis by macrophage and neutrophil. IgG activate antibody-dependent cell mediated cytotoxic mediated by natural killer cell. We have neonatal immunity, that's transfer of maternal antibody across the placenta and guts. Feedback inhibition of B cell activation, it functions that way. Also, it's neutralization of microbes and toxin. IgM is for activation of classical pathway for complement, also for neutralization of microbes and toxin. IgA is mucosal immunity, secreted by a secretion of IgA into lumen of gastrointestinal or respiratory tract. It is also for neutralization of microbes and toxin in the lumen and mucosa. IgE is most cell degranulation, immediate hypersensitivity reaction, and defense against the Helminth. So the effectors of function of antibody that have been mediated by Fc region have triggered the binding of antigen to variable region. The binding of antibodies to a multivariant antigen, such as polysaccharide or repeated epitope on a microbial surface, bring multiple antibody molecules close together, and this clustering of antibody molecules lead to the complement activation and allow antibody to bind to and activate Fc receptor and phagocytosis. The requirement for antigen binding ensure that antibodies activate virus effector mechanism only when they are needed. That is, when the antibody encounter and specifically bind antigen, not when the antibody are circulating in antigen-free forms. With this introduction to humoral immunity, we proceed to discussion of various function of antibodies in host defense. Neutralization of microbes and microbial toxin. Antibody against microbes and microbial toxin blocks the binding of these microbes and toxin to the cellular receptor. In this way, antibody inhibit or neutralize the infectivity of microbes as well as the potential injury effects of microbial toxin. Many microbes enter the host cell by the binding of particular microbial surface molecule to membrane protein or lipid on the surface of host cell. For example, influenza virus uses the envelope hemagglutinin to infect the respiratory epithelial cells, and gram-negative bacteria use pill to attack to and infect the variety of host cells. Antibodies that bind to this microbial structure interfere with the ability of the microbes to interact with the cellular receptor by means of steric hindrance, and thus prevent infection. Many microbial toxins mediate the prolonged pathological effect also by binding to specific cellular receptors. For instance, tetanus toxin bind to the receptor in the motor ends plates of neuromuscular junction and inhibit neuromuscular transmission, which lead to the paralysis, and diphtheria toxin bind to the cellular receptor and enter the virus cells, where it inhibits the protein synthesis. Antibodies against such toxin sterically hinder the interaction of the toxin with host cell, and thus prevent the toxin from causing the tissue injuries and disease. Neutralization can occur in multiple ways that go beyond steric interference. For instance, in lumen of the gut, aggregation or agglutination of microbes by IgA antibodies can reduce the infectivity of pathogen trap them in mucus and facilitate their clearance by peristalsis. In some cases, antibodies may bind to a microbes and induce conformational change in surface molecule that prevent microbes from interaction with the cellular receptor. Such interaction have been observed with antibody against certain viruses and are examples of historic effect of antibodies. Antibody-mediated neutralization of microbes or toxins require only the antigen binding region of the antibodies. Therefore, such neutralization may be mediated by antibodies of any isotopes in the circulation and in mucosal secretion, and can experimentally or therapeutically also be mediated by a Fab or Fab bracket AB2 fragment specific antibody which lack the Fc region of heavy chains. Neutralizing antibodies in the blood are mainly of IgG isotype and mainly IgA antibodies at mucosa site. The most effective neutralizing antibodies are those with high affinity for their antigen. High affinity antibodies are produced by process of affinity maturation. Many of prophylactic vaccine works by stimulating the production of high affinity neutralizing antibodies. A mechanism that microbes have developed to evade host immunity is to mutate the gene encoding surface antigen that are targets of neutralizing antibodies. For the role of antibodies in neutralizing microbes and toxic particular anatomic site, specifically in mucosal tissue and fetus, it is discussed in chapter 14. Antibody-mediated opsonization and phagocytosis. IgG antibody coats or opsonize microbes and promote their phagocytosis by binding to Fc receptor of phagocytes. Mononuclear phagocytes and neutrophil ingest microbes as prelude to intracellular killing and degradation. These phagocytes expresses a variety of surface receptor that directly bind microbes and ingest them, and even without antibodies, which are mechanism of innate immunity. The efficiency of this process is markedly enhanced with phagocytes if the phagocyte can bind the particle with high affinity. Mononuclear phagocytes and neutrophil expresses receptor for Fc portion of IgG antibodies that specifically bind antibody-coated particles. Microbes may also be coated by products of complement activation called C3b and then phagocytes by binding to leukocyte receptor for C3b described later in this chapter. As discussed in chapter 4, a process of coating particle to promote their phagocytosis is called opsonization. And the process and the substance that perform this function, including antibodies, complement protein, and certain plasma lectin, are called opsins. So this is the grammatical presentation. We see neutralization of microbes and toxin by antibodies. A here, this is all about antibody prevent binding of microbe to cells and thus block the ability of the microbes to infect cell. Or B, antibody inhibit the spread of microbes from infected cell to adjacent an infected cell. So the receptor for microbes, infected tissue. So if it is binded, now it is taken inside, so it cannot bind. C is antibody block the binding of toxin cells and thus inhibit pathological things like you see cell surface receptor of toxin, pathological effect toxin e.g. and cell necrosis, so it's inhibited. Leukocytes Fc receptor. Leukocyte expresses Fc receptor that bind to a constant region of antibodies and thereby promote the phagocytosis of Ig-coated particle and deliver signal that regulate activities of leukocyte. Other Fc receptor mediate the transport of antibodies to various sites. Fc receptor for different Ig heavy chain isotopes are expressed on many leukocytes population and serve a diverse function in immunity. Of these Fc receptor, the one that are the most important phagocytosis of opsonizing particle are specific for heavy chain Ig antibodies called Fc gamma receptor, and these are the receptor that will be primarily considered in this chapter. In chapter 20, we will discuss the Fc receptor that bind to IgE. In chapter 5, we describe the neonatal Fc receptor, which is expressed in placenta and vesicular endothelial and other cell types and has unique function related to the Ig transport across placenta and protection of IgG from a turnover. In chapter 14, we will discuss the poly-Ig receptor, which is involved in transport of mainly IgA across mucosal epithelia. Fc gamma receptor have been classified into three groups based on their affinity to heavy chains of different Ig subclasses. Different Fc receptor are also expressed there in different types. In general, IgG1 and IgG3 containing immunocomplex bind efficiently to activating Fc receptor, and IgG2 containing complex do not bind well. IgG4 has a very low affinity for activating Fc receptor, and the biological function of this antibody isotope is poorly understood. All Fc receptors are optimal activated by antibodies and antibodies bound to the antigen and not free circulating antibodies. The binding of immune complexes to most Fc receptor results in cellular activation except Fc gamma RIIB, which is an inhibitory receptor. All Fc gamma receptor contain ligand binding chain called an alpha chains that recognize IgG heavy chains. Difference in specificities or affinities of each FcR for various IgG isotopes are based on differences in the structure of these alpha chains. In all FcRs except FcRII, the alpha chain is associated with one or more additional polypeptide chain involved in signal transduction. Signaling function of all form of Fc gamma RII are mediated by cytoplasmic tail of this single chain receptor. So there is a table here talking of the Fc receptor. So Fc receptor gamma R1, this is mostly called CD64 as well, and it is mostly available in macrophage, neutrophil, and eosinophil. Function is phagocytosis, activation of the phagocytes. We have Fc gamma RIIA. This is CD20. This is CD32. It's low KD and it is mostly available in macrophage, neutrophil, dendritic cell, eosinophil, and is for phagocytosis cell activation. We have Fc gamma RIIB, which is CD32 as well, and it's low and it's mostly expressed in B lymphocyte, macrophage, dendritic cells, and other cells. Feedback inhibition for various cellular response. Then we have FcRIIC. This is a macrophage neutrophil and is functioning for phagocytosis cell activation. We have Fc gamma R111, IIIA. This is a CD16 and it's mostly available in neutrophil cells, macrophage, and dendritic cell. The antibody-dependent cell-mediated cytotoxic natural killer cell. We have FcRIIB. This is found in neutrophil phagocytosis initiations. We have Fc epsilon RI, that is mostly found in mast cell and for cell activation degranulations. Fc epsilon RII, this is CD23, mostly found in B lymphocyte, eosinophil, Langerhans cells, and the function is still unknown. And Fc alpha R, This again found in neutrophil, eosinophil, monocyte, and thought that it is causing cell activation. Therefore, there are three major groups of IgG-specific Fc receptor, of which two have multiple isoforms that differ in structure and function, and these are described in the following list. The unique function of FcRn are discussed in Chapter 5 and Chapter 14. Fc gamma Ri, which is CD64, is the major phagocyte Fc receptor. It is expressed on the macrophage and the neutrophil and bind to IgG1 and IgG3 with the high affinity dissociation constant of 10 power negative 8, 10 to 10 power negative 9. The larger extracellular amino terminal region of the Fc binding alpha chain folded into three tandem Ig-like domain. The alpha chain of FcRi is associated with the disulfide-linked homodimer of a signaling protein called FcR gamma chain. This gamma chain is also found in signaling complexes associated with the Fc gamma RII, Fc alpha R, Fc epsilon Ri. The gamma chain has only short extracellular amino terminals but larger cytoplasmic carboxyl terminus, which is structurally homologous to zeta chains of the T cell receptor complex. Like the T cell receptor zeta chain, the Fc R gamma chain containing immunoreceptor tyrosine based activation motifs that couples receptor clustering to activation of protein tyrosine kinase. A cross-linking of Fc receptor bond IgG molecule by multivariant antigen results in cell activation. Fc gamma RIIs, which is equivalent to CD32, bind IgG and IgG3 with low affinity, with the dissociation constant of 10 power 6. Genes duplication and diversification have resulted in the generation of three forms of this receptor called Fc gamma RII A, B, and C. These isoforms have similar extracellular domain and ligand specificity, but differ in cytoplasmic tail structure. The distribution and function of Fc RRIIA is expressed on neutrophil and mononuclear phagocytes and dendritic cell and participate in phagocytosis of opsonized particles, whereas Fc gamma RIIC is expressed in mononuclear phagocyte, neutrophil, and natural killer cells. The cytoplasmic tail of Fc RIIA and Fc RIIC contain items which on crossing IgG or IgG1 or IgG3 coated particles or cell can deliver an activation signals to phagocyte. On dendritic cell with receptor can contribute to capture the antigen in immune complexes and consequently T cell activation. Fc gamma RIIB is an inhibitory receptor expressed on myeloid cells and B cell, and it is the only Fc receptor on the B cells. Its role antibody feedback described in chapter 11. Fc RIII, which is equivalent to CD16, is also a low affinity receptor for Ig. The extracellular ligand binding portion of Fc RII is similar to Fc RII in structure, affinity, and specificity for IgG. This receptor exists in two forms, encoded by separate genes. The Fc gamma RIIA isoform is the transmembrane protein expressed mainly on natural killer cells and also macrophage, dendritic cell. Fc gamma RIIA associated with the homodimer of Fc gamma chain, homodimer of T cell receptor zeta chains. The associated chain contain items for that deliver activating signal upon antibody binding to Fc region, and thus necessary for function of receptor for receptor. The Fc RIIB isoform is glycophosphatidyllinositol linked protein expressed on neutrophil. It does not mediate phagocytosis or trigger neutrophil activation, and its function is poorly understood. In addition to this Fc receptor, there are receptor for heavy chain, IgE, IgA. We will describe the FcERI in chapter 20. The function of Fc alpha R is not well established. So these are figure 13.3, and we see subunity composition of Fc receptors, schematic model of different human Fc receptors illustrate the Fc binding alpha chains and signaling subunity. Fc RIIB is glycophosphatidyllinositol anchored membrane protein with no known signaling function. Fc RIIA and the IIC are structurally similar low affinity activating receptor with high different pattern of expression. Note that although Fc gamma RIIA/C and Fc gamma RIIB are both designed for CD32, they are different in protein with distinct functions. See the text. The neonatal FcR resemble class 1 major histocompatibility complex molecule structurally, but not have a peptide binding cleft Ig. So this was just structure. High affinity Fc gamma RI. This is low affinity, the Fc gamma RII2, that's A of C. Again, this is low affinity, but Fc gamma RIIB is used as inhibitor. So it's used as inhibitor. That's the main difference that you're going to see. Role of Fc receptor in phagocytosis and activation of phagocyte. Binding of Fc receptor on phagocytes to particle coated with antibody molecule lead to the engulfment of particle and activation of phagocytes. The Ig subtypes that bind best for Fc gamma receptor are Ig1 and Ig3 are the most efficient options for promoting phagocytosis. As discussed earlier, Fc gamma R1 is the high affinity Fc receptor on phagocytic cell and is the most important receptor for phagocytosis and opsonized particles. Opsonized particles are internalized into vesicle through a phagocytosome which fuse with lysosome, and the phagocytosed particles are destroyed in these phagolysosomes. Activation requires a cross-linking of FcRs by several adjacent Ig molecules, e.g. on antibody-coated microbes or immune complexes. A cross-linking of the ligands binding alpha chain of FcR result in signal transduction events that are similar to those that occur after antigen receptor cross-linking into lymphocytes. These include SRC kinase mediated tyrosine phosphorylation of items in the signaling chains of FcRs, SH2 domains mediated recruitment of SYK family kinase to items. Activation of phosphatidyl inostols 3 kinase recruitment of adapter molecule include SLP76 and BLNK, and recruitment of enzymes such as phospholipase, C gamma and the Tec family kinase. These events lead to the generation of ionostols, triphosphate, and diacylglycerol, and sustained increase in cytosolic calcium. Signaling pathway downstream of Fc gamma receptor induce a number of responses in leukocyte, including transcriptions of the genes encoding the cytokines, inflammatory mediator, and microcidal enzymes, and mobilization of the cytoskeleton, leading to phagocytosis, granular exocytosis, and cell migrations. The major microcidal substance produced in the activated phagocytes are reactive oxygen species, nitric oxide, and hydrolytic enzymes. These are the same substances produced by phagocyte activated in the innate immune response discussed in chapter 4. These microbial substances may also damage tissue. This mechanism of antibody-mediated tissue injury is important in hypersensitivity disease that we are going to look in chapter 18. Knockout mice lacking the ligand binding alpha chains FcR or the signal transducing FcR gamma chain are defective in antibody-mediated defense against microbes and do not develop some form of IgG antibody-mediated tissue injury, thus demonstrating the essential role of Fc receptor in this process. So we have the diagram here. So antibody mediated opsonization and phagocytosis in microbes. The antibody of certain immunoglobulin G, IgG subclasses bind to microbes and are then recognized by Fc receptor on phagocytes. Signal from the Fc receptor promote the phagocytosis of the opsonizing microbes and activate the phagocytes to destroy these microbes. The microcidal mechanism of phagocytosis are described in chapter 4 and with other figures. So here we see opsonization of microbes by IgG. After being opsonized, then we see phagocytes with the Fc gamma R1 that is pending, then binding of opsonized microbes to phagocyte Fc receptor. Then after binding it, you see yes, then Fc receptor signal activate phagocytes. Then the phagocytosis of microbes happens and killing ingested microbes happen. Inhibitory signaling by Fc gamma RII beta receptor. The Fc RII beta receptor is an inhibitory Fc receptor that was described earlier in the context of inhibitory signaling in B cell and the phenomena of antibody feedback. The Fc gamma RIIB is also expressed on dendritic cell, neutrophil and macrophage and mast cell, and may play a role in regulating the response of these cells to activating Fc receptor and other stimuli. A somewhat empirical but often a useful treatment of several autoimmune and inflammatory disease is intravenous administration of pooled human IgG called intravenous immunoglobulin IVIG. IVIG may bind to Fc gamma RIIB and deliver inhibitory signals to B lymphocytes and myeloid cells, thus reducing antibody production and dampening inflammation. However, numerous other mechanisms have been proposed to explain how IVIG work. Antibody-dependent cell-mediated cytotoxics, cytotoxicity. Natural killer cell and other leukocytes bind to antibody-coated cells by Fc receptor and destroy these cells. This process is called antibody-dependent cell-mediated cytotoxicity, ADCC. It was first described as the function of natural killer cell which uses the Fc receptor. Fc gamma RIIA, which is equivalent to CD 16 to bind to antibody-coated cells. Fc gamma RIIIA is a low affinity receptor that binds clustered IgG molecule displayed on the cell surface but does not bind circulating monomeric IgG. Therefore, antibody-dependent cell-mediated cytotoxic occur only when target cell is coated with antibody molecules, and free IgG in plasma neither activate natural killer cell nor compete effectively with cell-bound IgG for binding to Fc RIII. Engagement of Fc gamma RIII by antibody-coated target cell activate the natural killer cell to synthesize and secrete cytokines such as interferon gamma, as well as to discharge the content of their granules, which mediate the killing function of this cell, seen already in chapter 4. Antigen-dependent cytotoxic T cells, cytotoxic antibody-dependent cell-mediated cytotoxis also can be mediated by macrophages. Antibody-dependent cell-mediated cytotoxicity can be readily demonstrated in vitro, but its role in host defense against microbes is not established. It may be mechanism for elimination of cells that are coated by certain therapeutic monoclonal antibodies, such as B cells and B cell-derived tumor cells that are targeted by anti-CD20 antibodies. Antibody-mediated clearance of Helminth. Antibodies, eosinophils, and mast cells function together to mediate the killing and expansion of some of Helminthic parasites. Helminth worms are too large to be engulfed by phagocytes, and their integuments are relatively resistant to microcidal products of neutrophil and macrophage. They can, however, be killed by toxic cationic proteins known as the major basic protein present in the granules of eosinophils. The immune response to Helminth parasite is dominated by T helper 2 cell activation, IgE antibody production, and eosinophilia, suggesting that all may cooperate in defense. Antibodies that coat Helminth, especially IgG, can bind to Fc receptor and on eosinophil and cause the degranulation of the eosinophils, releasing cationic granules, proteins, and other mediators that kill the parasite. Neither human nor mouse eosinophil expresses appreciable level of high affinity IgE receptor, and in human eosinophil this receptor lacks the signaling beta chains, so a direct role of IgE in activating these cells seem unlikely. Human eosinophil can express a low affinity IgE receptor, CD23, but the function of CD23 in these cells remain poorly understood. IgE antibody that recognize antigens on the surface of the Helminth may also initiate local mast cell or basophil degranulation through the high tight affinity of IgE receptor. See in the figure in chapter 20. Mast cell mediator can induce bronchoconstriction and increase intestinal motility, contributing to the expansion of worms from sites such as airway and lumen of gastrointestinal tract. So here we have a diagram. So antibody-dependent cell-mediated cytotoxics, antibodies of certain immunoglobulin G subclasses bind to the cell, e.g. infected cell, and Fc region of the bound antibody are recognized by Fc gamma receptor on natural killer cells. NSA cells are activated and kill antibody-coated cells. So we see IgG has binded with surface antigen, antibody-coated cells. So this one, what will happen is Fc RII and natural killer cell. This one, it will kill. This killing of antibody will be happening because it's already coated. Now, let us start a long journey of complement system. and I think it will be joyous. The complement system is one of the major effector mechanisms of humoral immunity and is also an important effector mechanism of innate immunity. We briefly discussed the role of complement in innate immunity in chapter 4. Here we will describe the activation and regulation of complement in more detail. The name complement is derived from the experiment performed by Jules Baudet shortly after discovery of antibodies. He demonstrated that if fresh serum containing an antibacterial antibody is added to the bacteria at physiological temperature 37.37 Celsius, the bacteria are lysed. If however the serum is heated to 56 Celsius or more, it loses its lytic capacity. This loss of lytic capacity is not due to decay of antibody activities, because antibodies are relatively heat stable and even heated serum is capable of agglutinating bacteria. The lytic capacity of serum could be restored by adding fresh serum, even from animal that had not been immunized. Baudet concluded that in order to lyse bacteria, serum must contain another heat labile component that is present in all individuals and assist a complement the lytic function of the antibody. This component was later given name complement. A complement system consists of serum and cell surface protein that interact with one another and with other molecules of immune system in highly regulated manner to generate a product that function to eliminate microbes. Complementary proteins are plasma proteins that are normally inactive. They are activated only under particular conditions to generate products that mediate various effector function. Several features of complement activation are essential for its normal function. Complement system is activated by microbes and by antibodies and lectin that are bound to microbes and other antigens. Thus, complement focuses immune attack on microbial surface. The mechanism of initial activation are described later. Activation of complement involves the sequential proteolysis of protein to generate enzymes complexes with the proteolytic activities. Protein that acquires proteolytic enzyme activities by the action of other proteins are called zymogens. The process of sequential zymogens activation and a defining features of proteolytic enzyme cascade is also characteristic of the coagulation and kidney system. Proteolytic cascade allow tremendous and rapid amplification because each active enzyme's molecule produced at one step can generate a multiple activated enzyme molecule at the next step. Several of the biological active cleavage product of complement activation become covalently attached to microbial surface to antibodies bound to microbes and other antigen and to apoptotic bodies. In the fluid phase, complement protein are inactive or only transiently active for seconds, but they become stably activated after they are attached to microbes, antibodies, or dying cell. Thus, the full activation and therefore the biological function of complement system are limited to microbial cell surface or to site of antibodies bound to antigens and do not occur in blood. Complement activation is inhibited by regulatory proteins that are present on normal host cells and absent from microbes. The regulatory proteins are an adaptation of normal cells that minimize a complementary mediated damage to host cell. Because microbes lack these regulatory proteins, complement activation can occur on microbial surface. Pathway of complement activation. There are three pathways of complement activation. The classical pathway, which is activated by IgM and IgG antibodies bound to antigen. The alternative pathway, which is activated on microbial cell surface in the absence of antibodies. and lectin pathway, which is activated by plasma lectin that binds surface carbohydrate on microbes. The names classical and alternative arose because of a classical pathway was discovered and characterized the first, but the alternative pathway is phylogenetically older. And alternative and the lectin pathway are effector mechanism of innate immunity, whereas the classical pathway is the major mechanism of adaptive humoral immunity. The central event in complement activation is proteolysis of complement protein C3 to generate biological active product and the subsequent covalent attachment of a product of C3 called C3b to microbial cell surface or to antibody bond to antigen. Although the pathway of complement activation differ in how they are initiated, all of them result in a cleavage of the most abundant complement protein, C3. Complement activation involves generation of proteolytic complex, the C3 converters, which cleave C3 into two fragments called C3a and C3b. By conversion, proteolytic product of each complement protein are identified by lower case letter, suffix A referred to as the smaller product and B refers to the large one. C2 is exceptional for historical reasons. C3b become covalently attached to the microbial surface or to antibody molecule bound to antigen. All of the biological function of complementary are dependent on the proteolytic cleavage of C3. For example, a complement activation promotes phagocytosis because C3b become covalently linked to microbes and phagocytes. Neutrophil on macrophage express receptor for C3b peptide produced by proteolytic of C3 and other complement protein stimulate the inflammation. In all three pathways of complement activation, after generation of C3b by the 3 convertase, a second enzyme complex called the C5 convertase is assembled, which cleave the C5 into C5a and C5b. The C5 convertase contribute both to inflammation and generation of C5a fragment and to the formation of pores in the membrane of microbial targets. The pathway of complement activation differ in how C3b is produced but follow a shared sequence reaction after cleavage of C5. With this background, we proceed to more detailed description of alternative, classical, and lectin pathway. So, in summary, we have figure 13.6, the L step of complement activation by alternative, classical, and lectin pathway. We have the classical, the alternative, classical, and lectin pathway. The alternative pathway is activated by C3b, binding to various activating surface such as microbial cell wall. The classical pathway is initiated by C1 binding to antigen antibody complex, and then lectin pathway is activated by a plasma lectin to microbes. The C3b that generated by action of C3 convertase bind to the microbial surface of antibody and become a component of enzyme that cleaves C5 convertase and initiate the late step of complementary activation. The late step of 3 pathway are the same, not shown, and complement activated by all 3 pathways serves the same function: mannose binding lectin associated serum proteins. So we go back to the diagram as we saw here. We have the alternative pathway. An alternative pathway, what is needed really is binding of complement protein to microbial surface. That's what is happening. Then we have the microbial surface here. We have C3b. This C3b surface is bind to the microbial surface, and C3 convertase is happening. This C3 convertase is will cleave the C to get C3A and C3B. C3B will come in, and C3B when bind with C3BB and cleavage. This C3B is like C5 convertase. Then C5 convertase, when broken down again, you raise the C5. So alternative pathway, actually you just need C3, and T3 is converted into C3B. Then T3B acts as T3 convertase that will work and continue. The classical pathway here involve, we need C1, there's C2 and others, but these ones they need the activation of this process to a point of forming the C3 convertase that will work for as normal. Then in the lectin pathway, the MASP, the mannose, it is called the MASP, is mannose binding lectin associated serine protein. So again, it's work with mannose, then it activates the mannose binding lectin, then it follows almost the same pathway. Let us go and see them individually. Alternative pathway. Alternative pathway of complementary activation result in proteolysis of C3 and stable attachment of its breakdown product C3b to microbial surface without a role of antibody. So normally C3 in plasma is being continuously hydrolyzed and then cleaved at low rate, 1 to 2% of the total plasma C3 per hour, to generate C3b in a process that is called C3 takeover. This process involves factor B and factor D described later. The C3 proteins contain a reactive thiester bond that is buried in the region of the proteins known as the thiester domain. When C3 is cleaved, the C3b molecule undergo a dramatic conformational change and the thiester domain flip out a large shift of approximately 85 angle, expose the previously hidden reactive thiester bond. A small amount of C3b may become covalently attached to the surface of the cell, including microbes, and through the thiester domain, which react with the amino acid or hydroxyl group of cell surface protein or polysaccharide to form amide or ester bonds. If these bonds are not formed, the C3b remain in fluid phase and the exposed reactive thiester bond is quickly hydrolyzed, rendering protein inactive. As a result, further complement activation cannot proceed in plasma. So in the normal fluid phase, we have C3 hydrolyzed, which is called takeover, and formation of T3 convertase. So this C3 is hydrolyzed and forming the T3 convertase with factor D and we need factor B and factor D. Then we get fluid phase T3 convertase. Then T3 convertase, after being formed, it leads to the T3 being converted into T3b and C3a. The T3b, when combined with other B factor which are there on surface of microbes with the factor B again, with factor D again, it forms the T3 convertase, which is stable. And this T3 convertase is when it cleaves, we get T3b, a lot of them. Then T3b covalently bind to the surface of T3b to form a C5 convertase, and C5 convertase, when it's there, then it cleaves the C5. So alternative pathway of complementary activation: spontaneous hydrolysis of plasma cells, plasma T3 leads to the formation of fluid phase C3 convertase and generation of CBB in a process called the C takeover. The activated hydrolyzed C3 indicates that C3 is deposited on the microbial surface where it binds factor B and form the alternative pathway T3 convertase. This convertase T3BB cleaves the C3 to produce more C3b, which bind to form microbial bind to the microbial surface and participate in formation of T5 convertase. The C5 convertases C3BB, C3b cleave the C5 to generate a C5b, the initiating event in the late step of complementary activation. So we have protein of alternative pathway complements. Protein C, this is involved. C binds. The main function is already just main function. Structure you'll have to go yourself if you want it. This is function is C3b binding to the surface of microbes, where it functions as an opson and as well as a component of C3 and C5 convertases. T3A stimulate inflammation, anaphylactosin. Then we have factor B. This is BB, is serine protease and the active enzymes of C3 and the C5 convertases. We have factor D. This is plasma serine protease cleavage factor B when bound to C3b. So it's a plasma serine protein cleavage factor B when bound to C. Then we have proedin. This is proedin stabilized C3 convertase, which is now complete C3BBB on microbial surface. When C3b undergoes its post-cleavage conformation change, a binding site of plasma protein called factor B is also exposed. Factor B then binds to C3b protein that is now covalently tethered to the surface of cell. Bound factor B is in turn cleaved by plasma serine protease called factor D, releasing a small fragment called BA and generating a large fragment of BB that remain attached to C3b. This is called C3b-BB complex in alternative pathway. T3b convertase the function to cleave more C3 molecule, thus setting up an amplification sequence. Even when C3b is generated by classical or lectin pathway, it can form complex with BB, and this complex is able to cleave more C3. Thus, the alternative pathway C3 convertase function to amplify a complement activation when it is initiated by any of the three pathways. When C3 is broken down, the T3b remain attached to cells and T3a is released. The soluble fragment has several biological activities that are discussed later. So in the normal way, we have intact C3 inaccessible thiostat group. They are cleaved by C3, the cleaves of C3 alpha chain, and they get to T3 convertase. So this process continue in the fluids, continue, then C3 the C3 is converted again, we get C3A. The whole process is happening. So internal thiostat bonds C3 proteolytic cleavage of alpha chains of C3 convert into the metastable form in which the internal thiostat bond are exposed and is susceptible to nucleophilic attack by oxygen atoms shown in nitrogen atom. The result is the formation of covalent bond with protein or carbohydrate on the cell surface. C4 is structurally homologous to C3 and has an identical thiostat bond. Okay, I think that one was new thing. Alternative pathway activation readily occur on microbial cell surface, but not on mammalian cells. If C3BBB complex is formed on mammalian cell, it is rapidly degraded and the reaction is terminated by the action of several regulatory proteins present in these cells, discussed later. Lack of regulatory proteins on microbial cells allow binding and activation of alternative pathway C3b convertase. In addition, another proteins of the alternative pathway called propedins can bind to and stabilize the C3BBB complex, and the attachment of propedin is favored on microbial as opposed to normal host cell. Some of the C3b molecule generated by alternative pathway, C3 convertase bind to the convertase itself. This results in the formation of complex containing 1b moiety and 2 molecular CB, which function as alternative pathway C5 convertases, which will cleave the C5 and initiate the latest step of complementary activation. Classical pathway. The classical pathway is initiated by binding of complementary protein C1 to C1 to CH2 domain of IgG or CH2 3 domain of IgM molecules that have bound antigen. So among IgG antibodies, IgG1 and IgG3 in human are more efficient activator of complement than other subclasses. IgG2 has some ability to activate complement, but IgG4 does not. C1 is a large multimeric proteins complex composed of C1q, C1r, C1s subunity. C1q bind to the antibody, and C1r and C1s are proteases. The C1q subunity is made up of an umbrella-like radio array of 6 chains, each of which has a globular head connected by a collagen-like arm to central staff. This hexamer performs the recognition function of the molecule and bind specifically to the Fc region of mu or some gamma heavy chain, as mentioned in chapter 4. C1q bind to pentraxin, such as C-reactive protein or serum amyloid protein, and also can bind to apoptotic bodies. Only antibody bind to antigen and not to free circulating antibodies can initiate classical pathway. The reason for this is that each C1q molecule must bind to at least 2 Ig heavy chain to be activated, and each IgFc region has only a single C1q binding site. Therefore, 2 or more Fc regions have to be accessible to C1 in order to initiate classical pathway activation. Because each IgG molecule has only one Fc region, multiple IgG molecules must be brought close together before C1q can bind, and multiple IgG antibodies are brought together only when they are simultaneously bind to identical epitope of multivariant antigens or to several antigens molecules on a microbe cells or tissue surface. Even though free circulating IgM is pentameric, it does not bind the C1q because the CH3 domain of 3M are in configuration that is inaccessible to C1q. Binding of the IgM to an antigen induces conformational change that exposes the C1q binding site in the CH3 domains and allow C1q to bind. Because of its pentameric structure, a single molecule of IgM can bind multiple CQ molecule, and it is one of the reasons that IgM is one of the efficient complementary binding, also called the complementary fixing antibody than IgG. C1r and C1s are serine proteases that form a tetramer containing 2 molecules of each protein. Binding of 2 or more of globulin head of C1qc to Fc region of Ig or IgM to enzymic activation of associated CR, which cleaves the activates C1s. Activated C1s cleave the next proteins in cascade, C4 to generate C4b. A small C4 fragment is released and has biological activity that are described later. C4 is homologous to C3, and C4b containing an internal thiouster bond similar to that of C3b that form covalent amide or ester linkage with a antibody complex or with adjacent surface of cell to which the antibodies bond. This attachment of C4b ensure that classical pathway activation proceed on the cell surface of immune complex. The next complementary protein is C2. Then complex with the cell surface bond C4b and is cleaved by the nearby C1s molecule to generate soluble C2b fragment of unknown importance and larger Ca fragment that remain physically associated with the C4b on the surface. The resulting is C4b2a complex in a classical pathway, F3 convertase. It has ability to bind to and proteolytically cleave C3, binding the enzyme complex to C3 mediated by a C4b component, and proteolysis is catalyzed by C2a component. Cleavage of C3 results in the removal of small C3a fragment, and C3b can form a covalent bond with cell surface on which antibody where complement activation was initiated. After C3b is deposited in, it can bind factor B and generate more C3b convertase by alternative pathway as discussed earlier. The net effect of multiple enzymic step and amplification is that millions of the molecule of C3b can be deposited within a minute on the cell surface where complement is activated. The key L step of alternative and classical pathway are analogous. C3 in the alternative pathway is homologous to C4 in classical pathway, and factor B is homologous to C2. Some of the C3b molecules generated by a classical pathway, C3 convertase bind to the convertase as a result in an alternative pathway and form C4 to C4 B2A C3b complex. This complex functions as a classical pathway C5 convertase. It cleaves the C5 and initiates the late step of complementary activation. Lectin pathway. Lectin pathway of complement activation is triggered by binding of microbial polysaccharide to circulating lectin such as plasma mannose binding lectin, or to ficolin. These soluble lectin are collagen-like protein These soluble lectin are collagen-like protein that structurally resemble C1q. See the figure 413. MBL L ficolin and F and H ficolin are plasma protein. M ficolin is mainly secreted by activated macrophage in tissue. MBL, mannose binding lectins, has N-terminal collagen-like domain and C-terminal carbohydrate recognition lectin domain, and thus a number of collagen family of serum agglutinin. The filicon has similar structure with N-terminal collagen-like domains and C-terminal fibrinogen-like domain. The collagen-like domain help to assemble basic triple helical structure that can form a higher order oligomers. Mannose binding lectin bind to mannose residue on polysaccharide and the fibrinogen-like domain of ficolin bind any acetylglucosamine containing glycan. These polysaccharides and glycan are abundant in bacteria and fungi. Both mannose binding lectins and the ficolin associate with mannose binding associated serine proteins MASP include MASP1, MASP2, MASP3. The MASP are structurally homologous to C1R and the C1s protease and serve a similar function, namely the cleavage of C4 and C2 to activate the complement pathway. Multimer of MBL associated with MASP1, MASP2, MASP3, and MASP2 is the protease that cleave C4 and C2. Subsequent events in this way are identical to those that occur in classical pathway. So binding of antibody multivariant antigen binding antibodies. So this is the classical pathway complement activation. Antigen antibody complex that activate the classical pathway may be soluble, fixed on the surface cell as shown, or deposited on extracellular matrix. The classical pathway is initiated by binding of C1 to antigen complex antibody molecule, which lead to the production of C3 and C5 convertases attached to the surface where the antibody was deposited. The C5 convertase cleave C5 to begin late step in complement system. Yeah, so binding of antibodies to multivariant antigen binding of C1 to antibodies. So these are antibodies, this is C1. What happened is this binding cleavage of C4 by C1 RS, C1 R and S enzymes, covalent attachment of then C4 is cleaved to get C4b to antigenic surface and antibodies. Thereafter, the cleavage will follow the cleavage of C2, binding of C2 to 4 to form C4b2A complex, which is 3 convertase. This one will do again, will do cleavage of 3 convertase, and then it will have more 3b. The binding of C3b to antigenic surface of C4b2A to form C4b2A3b complex, which is 5 convertase, which is now it continued for other component. Protein of classical pathway. So in the protein of classical pathway we have C1q. The main function is bind to the Fc portion of antibody that has bound antigen to apoptotic cell and to the cationic surface. We have C1R, this is serine proteins, cleave C1s to make it an active protease. This is serine protease that cleaves C4 and C2, that's called C1s. C4, this is C4b covalently bond to the surface of microbes or cell where the antibody is bound and complement is activated. C4b again bind to C2, form the cleavage of C1s. C4a stimulate inflammation and Phagocytosis. C2, this is serine proteins that function as active enzymes of C3 and C5 convertase to cleave the C3 and the C5. So we have a late step of complementary activation. So C5 convertases generated by alternative classical or lectin pathway initiate activation of the late component of complementary system, which culminate to formation of a cytosol membrane attacking complex MAC. The C convertase cleave the C5 into the small C5a fragment that is released and a two-chain C5b fragment containing alpha and beta chain. That is also released by bind rapidly to plasma C4 at C6. C6 undergoes the conformation change and the C5b6 complex then bind to the cell membrane through both an ionic and hydrophobic interaction. C5a has potent biological effect on several cells and are discussed later. C7 from plasma then bind the chain of C5b and form the C5b, C6, C7 then complex. The bond of C5 undergoes amphiphilic transition and penetrate the membrane. It can contribute to the release of some phospholipid micellin from the membrane but does not form the complete pores. C8 protein is trimer composed of three distinct chains, one of which bind to C5b component of the C5b to 7 complex and form a covalent heterodimer with a second chain. The third chain insert into the lipid bilayer of the membrane. This stably inserted C5b678 complex form unstable poles that range from 0.0 to 3 nanometer diameter, and a very large number of these C5b to 8 complex annulized cells. The formation of full active MAC is accomplished by binding of C9, the final component of complementary cascade to C5b8 complex. C9 is serum protein that polymerizes at the site of bond to form pores in plasma membrane that are made up of C5b9 complex containing of C5b6, 7, 8 and many molecules of C9. These pores are approximately 20 nanometer in external diameter, 1 to 12 to 11 nanometer in internal diameter, have height of approximately 15 nanometer and form channels that allow free movement of water and ion. The channel size, based on the number of C9 molecule in the C5b to C9 complex, tubular complex of C9 alone may also form. The entry of water results in osmotic swelling and rupture of the cells on those surface of MAC is deposited. The pore formed by polarized C9 are similar to the membrane pore formed in perforin and cytoplasmic granule proteins found in cytotoxic T lymphocyte and natural killer cell, and C9 is structurally homologous to perforin. So this is the complex. We see structure of C1 C1q consists of 6 identical subunit arrangement from the central cores and is symmetrically projecting radial arm. The globular head of at the end of each arm designated the H the contact region, the FC portion antibodies, IgG in the case. The C1R and C1S form a tetramer composed of 2 C1R and 2 C1S molecules. The end of C1R, C1S containing catalytic domain of this protein. 1 C1R 2 and S2 tetramer wrap around the radial arms of the C1q complex in a manner that juxtaposes the catalytic domain of C1R C1S. Complement activation. Soluble IgG exposure not adjusted. If they are not collected together, they cannot form. But if they are together, they can activate them. As we are told earlier that one cannot activate. So it cannot work. Those are things that you have to remember. So proteins of the lectin pathway complement. So in lectin pathway complement, we have protein mannose binding lectin. This is main function is agglutinin, opsinin, with the other with complement fixing. We have M-ficolin, ficolin 1. The function is agglutinin, opsinin, complement fixing. We have L-ficolin. Again, the function is agglutinin, opsinin, complement fixing. We have H-ficolin, ficolin 3. The main function is agglutinin, opsinin, and complex fixing. We have MASP. This forms the complex with MASP2 and collectin and O-ficolin and activate MASP3. MASP2 form complex with eclin, especially ficolin 3. MASP3 associate with a collectin or ficolin and MASP1 and cleavage C4. Protein of late step complement activation. C5 is a C5b initiate assembly of MAC. C5a stimulate inflammation and aflatoxin. Then is C6 component of MAC bind to C5b and accept C7. C7 component of MAC bind to C5b as C5b C6 and insert into lipid membrane. Then H component of MAC again and initiate the binding and polymerization of C9. C9 is a component of MAC bind to C5b, 6, 7, 8 and polymerize to form membrane pores. So those are things that we are trying to speak. So I think we go direct to receptor for complement proteins. 1 minute. So receptor for complement proteins. Many of biological activities of complement system mediated by the binding of complement fragment to membrane receptor expressed on various cell types. The best characterized of these receptors are specific for fragment of C3 and are described here. So type 1 complement receptors CR1s or CD35 function mainly to promote phagocytosis of C3b and C4b coated particles and clearance of immune complex from circulation. CR1 is high affinity receptor for C3b and C4b. It is expressed mainly on bone marrow derived cells including erythrocyte, neutrophil, monocyte, macrophage, eosinophil, and T and B cells. It is also found in follicular dendritic cells in a follicle of secondary lymphoid organs. Phagocytes use this receptor to bind on the internalized particles opsonized with C3b and C4b. Binding of C3b or C4b coated particle to CR1 also transduces signal that activate the microcidal mechanism of phagocyte, especially when Fc receptor is simultaneously engaged by antibody-coated particles. SR1 on and on erythrocytes bind circulating immune complex with the attached C3b and C4b and transport the complex to the liver and spleen. Here, phagocyte removes the immune complex from erythrocyte surface and erythrocyte continue to circulate. CR1 is also a regulator of complement activation discussed in the section that follows. Type 2 complement receptor CR2 or CR21 function to stimulate humoral immune response by enhancing B cells activation by antigen and by promoting the trapping of antigen antibody complex in germinal center. Complement receptor 2 is present on B cell lymphocyte, follicular dendritic cells, and some epithelial cell. It specifically binds cleavage product of C3b called the C3d, T3DGs, and the IC3b. The I refers to inactive, which are generated by factor 1 mediated proteolysis discussed later on B cells. Complement receptor 2 is expressed as part of trimolecular complex that include two other non-covalent attached protein called CD19 and CD81. TAPA1, target of anti-proliferative antibody 1. This complex delivers signal to B cells that enhance the response of B cell to antigens. On follicular dendritic cells, complement receptor 2 serves to trap I inactive C3b, C3d, and C3dg coated antigen antibody complex in germinal center. The function of complement in B cell activation are described later. The type 3 complement receptor, also called MAC1 or CR3, CD11, BC, CD8, is an antigen that is an integrin, not antigen, an integrin that functions as a receptor for ICB fragment produced by proteolysis of C3b. MAC is expressed on neutrophil, mononuclear phagocyte, mast cell, and natural killer cells. This member of integrin family consists of a chain alpha chains CD11b covalently linked to alpha B chain that is identical to beta chains of two closely related integrin molecules. LFA, leukocyte function associated antigen, and P15090, which is CR4, MAC1 on neutrophil and monocytes promote phagocytosis of microbes opsonized in IC3b. In addition, MAC1 may directly recognize bacteria for phagocytosis by binding to some unknown microbial molecules. It also binds the ICAM, intercellular adhesion molecule 1, on endothelial cells and promote stable attachment of leukocyte to endothelium even without complement activation. This binding leads to the recruitment of leukocytes to sites of infection and tissue injuries. Receptors for fragment of C3. Type 1 complement receptor. The main function is phagocytosis, clearance of immune complexes, promotes dissociation of T3 converters, and the cofactors for cleavage. There are many others like type 2 complement receptor. They are co-receptor for B and activation. They work for activation. They are also responsible for trapping of antigen in the immunocenter. T3 complement, which is equivalent to T3, MAC1, CD11B, CD18, and is functioning for phagocytosis, leukocytizations, endothelium via ICAM. Type 4 complement also for phagocytosis and adhesions molecule, but they are querying. So CCRP, the complement control proteins, C. Epstein-Barr virus, C. follicular dendritic cell, those are just the description in these places. So type 4 complement receptor, this is CR4, which is equal to P5095 CD11C CD18, is another integrin with different alpha chains, CD11C, and the same B chains as MAC1. It also binds to inactive C3b, and the function of this receptor is probably similar to the MAC1. CD11C is abundantly expressed in the dendritic cell and is used as the marker for this type of cell, CD11C. Complement receptor of immunoglobulin family. Complement receptor of immunoglobulin family is expressed on surface of macrophage in the liver known as Kupffer cell. So complement receptor for immunoglobulin G is an integral membrane protein with an extracellular region made up of Ig domain. It binds complement fragment 3C3b and IC3b, and it's involved in the clearance of opsonized bacteria and other blood-borne pathogens. Other receptors include those of C3a, C4a, C5a, which stimulate inflammation, and for C1q, the pro-inflammatory effects of C3a, C4a, C5a are mediated by binding these fragments to specific receptor on various cell types. The C5a receptor, A receptor is the most thoroughly characterized. It is the member of G protein-coupled receptor family expressed on many cell types, including neutrophil, eosinophil, basophil, monocyte, macrophage, mast cell, endothelial cells, smooth muscle cell, and epithelial cell, and astrocytes. The C3A receptor is also a member of G protein-coupled receptor. This receptor collaborates with Toll-like receptor in the activation of sentinel cells of phagocytes. This C1q receptor is expressed in phagocytes and may assist in the clearance of apoptotic bodies and protein fibers, much as myeloid fibril, both which bind the C1q. Regulation of complement activation. Activation of complement cascade and the stability of active complement proteins are tightly regulated to prevent complementary activation on normal host cell and to limit the duration of complement activation even on microbial cells and antigen-antibody complex. Regulation of complement is mediated by several circulating cell membrane proteins. Many of these proteins belong to the family called regulator of complement activity and are encoded by homologous genes that are located adjacent to another one another on chromosomes 1 and Q2,3 RCA. Protein include this membrane protein decay accelerating factor CD55, membrane cofactor protein CD46, complement receptor 1 and complement receptor 2. The circulating RCA proteins in plasma include factor H and C4 binding proteins. Complement activation needs to be regulated for two reasons. First, low level of complement activation goes on spontaneously, and if such activation is allowed to proceed, the result can be damage to the normal cells and tissues. Second, even when complement is activated where needed, such as on microbial cell, the antigen-antibody complex is needed. It needs to be controlled because the degradations products of the complement protein can diffuse to adjacent cell and injure them. So these are regulatory complement activations, and we have C1 inhibitor. See, this function is serine protease inhibitor binding to C1R and C1SC, dissociate them from C1q. We have factor 1 that is mostly interact with C4b, C3b, and is found in serine function is in serine protease cleave the C3b and C4b by using factors H, Mcp, C4b, CRI cofactor. Factor H is found in is interacting with the C3b and bind with the C3b to displace BP. Cofactor for factor 1 mediated cleavage C3b. For C4 binding proteins, it is interacting with its C4b and bind to C4b, displace C2. There are membrane cofactor proteins that are interact with C3b, C4b, the cofactor for factor I mediated cleavage of C3b, C4b. Decay accelerated factor, they interact with C4 to C4b, 2a, C3b, bb, and they displace the C2a's from C2b and bb from CB, dissociation. Well, we have CD59 that's interact with with C7 and C8, block the C9 binding, prevent the formation of MAC. So different regulatory mechanisms inhibit formation of C3 convertase in early step of complement activation, breakdown and inactivate C3 and C5 convertase, and inhibit formation of MAC in later step of complement activation. So proteolytic activities of C1R, C1S, and MASP-P is inhibited by plasma protein called C1 inhibitor. C1 inhibitor is the serine protease inhibitor, sepin, that mimic the normal substrate of C1R, C1S. If C1q bind to an antibody and begin the process of complement activation, C1 inhibitors becomes a target for the enzymatic activity of bound C1R, C1S. C1 inhibitor is cleaved by and become covalently attached to this complement protein, and as the result of C1R and C1S2 tetramer dissociate from C1q, thus stopping activation by classical pathway. In this way, C1 inhibitors prevent the accumulation of enzymatically active C1R, C1S in plasma and limit the time for which the active C1R, C1S is available to activate subsequent steps in complement cascade. Similarly, by inactivating the MASP2, C1 inhibitor also dampen the lectin pathway, an autosomal dominant inherited disease called hereditary angioedema. The angioedema is due to deficiency of C1 inhibitor, or C, it is called inhibited by plasma protein called C1 inhibitor. Clinical manifestation of disease include intermittent acute accumulation of edema fluid in the skin and mucosa, which causes abdominal pain, vomiting, diarrhea, potential life-threatening airway obstruction. In some of these patients, plasma level of C1 inhibitor protein are sufficiently reduced, less than 20% to 30% of normal. That activation of C1 by immune complexes is not properly controlled, and increased breakdown of C4 and C2 occurs. The mediator of edema formation in patients with hereditary angioedema include proteolytically fragment of C2 called C2 kinin and bradykinin. C1 inhibitors is an inhibitor of other plasma serine proteins beside C1, including kallikrein and coagulation factor 12, both of which they can promote increased formation of bradykinin. Recombinant of C1 inhibitor is now used to treat the patient with this deficient. Assembly of component of C3 and C5 convertase is inhibited by binding of regulatory protein of RCA family to C3b and C4b deposited on cell surface. If C3b is deposited on surface of normal mammalian cells, it may bound by several membrane proteins, including MCP, CD45, CR1, DAF, and the plasma protein H. C4b deposited on cell surface is similarly bound to DAF and CR1, MCP, and another plasma protein C4PB by binding to C3b and C4b. This protein competitively inhibit the binding of other component of C3b converters, such as BB of alternative pathway and the C2 of the classical pathway, thus not blocking the further progression of complement cascade. Factor H inhibits binding of only BB to CB and thus regulators of the alternative but not the classical pathway. MCP, CR1, and DAF are produced by mammalian cells but not the microbes. Therefore, these regulators are regulators of complementary selective inhibitors complement activation on host cell and allow complement activation to proceed on microbes. In addition, cell surface rich in sialic acid favor in binding of regulatory protein factor H over the alternative pathway proteins. Factor B, mammalian cells expresses high level of sialic acid than most microbes do, which is another reason for that the complementary activation is prevented on normal cells and permitted on the microbes. DAF is a GPI-linked membrane protein expressed on the endothelial cell and the erythrocytes. A hematopoietic stem cell deficiency of enzymes required to form such protein lipid linkage result in lack of blood cells expression of many GPs-linked membrane protein, including DAF and the CD59. See later. Cause the disease called the proximal nocturnal hemoglobinuria. This disease is characterized by recurrent bout of intravesicular hemolysis, at least partly attributable to unregulated complement activation on the surface of erythrocytes. Recurrent intravesicular hemolysis in turn lead to the chronic hemolytic anemia and venous thrombosis. An unusual features of this disease is that the causative mutation in the gene that encode an enzyme responsible for the generation of GPI anchor is not inherited, but it is acquired somatic mutation in a hematopoietic stem cell. A rare inherited deficiency of DAF causes small vessel thrombosis and protein losing enteropathy, which respond well to a C5 inhibitor. Thereby, it does not cause paroxysmal nocturnal hemoglobinuria because of CD59 expression is normal. So these are structure that you can read. These are the regulation of activities, regression activity. Cell-associated C3b is proteolytically degraded by a plasma serine protease called factor 1, which is active only in presence of regulatory proteins. MCP, factor H, C4BP, and CR1 all serve as cofactor for factor 1 mediated cleavage of C3b and C4b. Thus, these regulatory host proteins promote proteolytic degradation of complement protein. As discussed earlier, the same regulatory protein causes dissociation of C3b and C4b containing complexes. Factor 1 mediated cleavage of C3b generate the fragment called IC3b, C3d, and C3dG, which do not participate in complement activation but are recognized by receptor on phagocyte and B lymphocytes. Inflammation induced by T3A and C5A is regulated by rapid cleavage of their C-terminal adenine residue by plasma carboxypeptidases. This results in generation of C3A -ag, C5A -ag, each of which has only approximately 10% of activity of native form of these proteins. Formation of the MAC is inhibited by membrane proteins called CD59. CD59 is GPI-linked protein expressed on the many cell types. It works by incorporating itself into assembly MAC after the membrane insertion of C5b8, thereby inhibiting subsequent addition of C9 molecules. C59 is present on normal host cell, there it's limiting the MAC formation, but it's not present in microbes. Formation of MAC is also inhibited by plasma proteins such as S protein, which function by binding soluble C5b67 complex and thereby preventing their insertion into cell membrane near the site where the complement cascade was initiated. Growing MAC can insert into any neighboring cell's membrane beside the membrane on which they were generated. Inhibitors of MAC is in the plasma and in host cell membrane ensure that lysis of innocent bystander cells does not occur nearby the site of complement activation. These are the diagrams that I cannot pass through now. Much of analysis of the function of complement regulatory protein has relied on in vitro experiment. Most of these experiments have focused on assay that measures the MAC-mediated lysis of erythrocyte as an endpoint. On the basis of these studies, hierarchy of importance of inhibitory complement activation is thought to be CD59 is greater than DAFI is greater than MASP. This hierarchy may reflect relatively abundance of these proteins on cell surface. So these are structure. Factor 1 mediated cleavage is C3b. Also in the presence of cell membrane bond cofactor, membrane cofactor protein, membrane cofactor protein MCP, a complement receptor CR1, plasma factor, proteolytic cleaves the C3b attached to the cell surface, leaving to the inactive form of C3b, IC3b. Factor H and C4 binding protein can also serve as cofactor for factor I mediated cleavage. The same process is involved in the proteolysis Yeah, again we see this one is regulatory formation of membrane attack complex. So all these are the regulated activation of component of complement. We have the formation of MAC here. So all these have to be controlled. The CD459 inhibit poly C9 assembly. We have S protein inhibit membrane insertion. So all these are happening in the normal range. So here the function of complement, the major function of the complement system is in the host as shown. The cell bound C3b is an opsin that promotes phagocytosis of coating cell. A, the proteolytic product of C5a, C5, C3a, and to a less extent C4a stimulate leukocyte recruitment and inflammation. B, the membrane attack complex. C is must be somewhere, but don't know where is it. Okay, so yeah, this is the structure. Optionalization and phagocytosis, stimulation of inflammation reactions, complement mediated cytocytosis. So this is MAC. This is the function of C3a, C3b, C5a. These are the function of other things. The function of regulatory protein may be overwhelmed by excessive activation of complement pathway. We have emphasized the importance of this regulatory protein in preventing complement activation on normal cell. However, complement-mediated phagocytosis and damage to normal cell are important pathogenic mechanisms in many immunological diseases. You'll see in chapter 19. In these diseases, large amount of antibody may be deposited on host cells, generating enough active complement protein that are regulatory molecule and are unable to control complement activation. Function of complement. The principal function of the complement system in innate immunity and adaptive humoral immunity are to promote phagocytosis of microbes on which complement is activated, to stimulate inflammation and to induce lysis of these microbes. So the main function of complement are the principal main function of complement system in innate immunity and adaptive humoral immunity are to promote phagocytosis of microbes on which complement is activated, to stimulate inflammation, and to induce cell lysis of these microbes. In addition, product of complement activations facilitate the activation of B lymphocytes and production of antibodies. Phagocytosis, inflammation, and the stimulation of humoral immunity are all mediated by the binding of proteolytic fragment of complement protein to various cell surface receptor, whereas cell lysis is mediated by MAC. In the following section, we will describe this function of the complement system and their role in host defense. Optionalization in phagocytosis. Microbes on which complement is activated become coated with the C3b, IC3b or CC4b and are phagocytosed by binding of this protein to specific receptor on macrophage and neutrophil. As discussed previously, activation of complement leads to the generation of C3b, IC3b covalently bound to cell surface. Both C3b and IC3b act as opsin by virtue of their ability to specifically bind to receptor of neutrophil and macrophage. C3b and C4b, later generated by classical pathway, only bind to CR1 and IC3b bind to CR3, MAC1 and CR4. By itself, CR1 is inefficient inducing the phagocytosis of C3b coated microbes, but its ability to do so is enhanced with the microbes if the microbes are coated with IgG antibody that simultaneously bind the Fc receptor. Macrophage activation by cytokine interferon gamma also enhances CR1 mediated phagocytosis. C3b and IC3b dependent phagocytosis of microorganisms is a major defense mechanism against infection in innate and adaptive immunity. One example of the importance of complement is host defense against bacteria with the polysaccharide-rich capsule, such as pneumococcus and meningococcal, which is mediated primarily by humoral immunity. IgM antibody against capsular polysaccharide bind to the bacteria, activate the classical pathway of complement and cause phagocytic clearance of bacteria in the spleen. This is why individuals lacking spleen as a result of autosplenectomy in sickle cell anemia patient are due to surgical removal after a traumatic rupture of the spleen or to treat the patient with autoimmune hemolytic anemia are susceptible to disseminated pneumococcal or meningococcal sepsemia. Stimulation of inflammatory response. The proteolytic complement fragment of C5a, C4a, C3a induce acute inflammation by activating mast cell, neutrophil, and endothelial cells. All three peptides bind to the mast cell and induce degranulation with the release of vasoactive mediators such as histamine. These peptides are also called anaphylaxon because the mast cells reactions they trigger are characterized by anaphylaxis. See chapter 20. In neutrophils, C5a stimulate motility, film adhesion to endothelial cells, and at high doses, stimulation of respiratory burst and production of reactive oxygen species. In addition, C5a may act directly on vesicular endothelial cells and induce increased vesicular permeability and the expression of P-selectin, which promote neutrophil binding. This combination of C5a action and mast cell, neutrophil, endothelial cell contribute to the inflammation at site of complement activation. C5a is the most potent mediator of the mast cell degranulation. C3a is the approximately 20 times fold less potent, and C4a is approximately 2,500 fold less. So the most potent is C5a. Complement-mediated cytolysis. Complement-mediated lysis of the foreign organism is mediated by Mc. Most pathogenic have evolved thick cell walls and capsules that impede access of the Mc to their cell membrane. Complement-mediated lysis appear to be critical for defense against only few pathogens that are unable to resist Mc insertion, such as bacteria of genus Neisseria, which have very thin cell walls. Other function of the complement system: by binding to antigen-antibody complex, complement proteins promote the solubilization of these complex and their clearance by phagocyte. Small number of immune complexes are frequently formed in circulation when an individual mounts vigorous antibody response in a circulating antigen. If the immune complexes accumulate in the blood, they may be deposited in vessel wall and lead to the inflammatory reaction that damage the vessels and the surrounding tissue. The formation of immune complex may require not only the multivariant binding of Ig-Fab region to antigen, but also non-covalent interaction of Fc regions of juxtaposed Ig molecules. Complement activation on Ig molecule can stereotypically block these Fc-Fc interaction, thereby promoting the dissolution of immune complexes. In addition, as discussed earlier, immune complexes with attached C3b bind to C1 CR1 on erythrocyte, and the complex are cleared by phagocytes in the liver. The C3d proteins generated from C3 bind to CR2 on B cell and facilitate B cell activation and development of humoral immune response. C3d is generated when complement is activated by antigen either directly, e.g. when the antigen is in antimicrobial polysaccharide, or after binding to antibody. Complement activation results to the covalent attachment of T3b and its cleavage product C3d to antigen. B lymphocyte can bind to antigen through their Ig receptor and simultaneously bind to the attached C3d through CR2, the core receptor on B cells, thus enhancing antigen-induced signaling in B lymphocyte. See chapter 7 and chapter 11. Opsonized antigens are also found by follicular dendritic cell in germinal center of lymphoid organs. Follicular dendritic display antigen to B cell in the germinal center, and this process is important for selection of high affinity B cells. The importance of complement in humoral immune response is illustrated by the impairment in the antibody production and germinal center formation seen in knockout mice lacking C3 or C4 or CR2 protein. Complement proteins and natural antibody contribute to the clearance of apoptotic cells and fragments. Many different host protein contribute to the clearance of apoptotic cells by macrophage, but the functional importance of complement protein in this process is revealed in patients with the inherited complement deficiency discussed next. Complement deficiency. Genetic deficiency of complement protein and regulatory proteins are the causes of various human disease inherited and spontaneous deficiency in many of the complement protein have been described in human. Genetic deficiency in classical pathway component, including C1q, C1R, C4, C2, C3, have been described. C2 deficiency is the most common human complement deficiency. More than 50% of the patients with C1q, C2q, C4 deficiency develop systemic lupus erythematosus. The reason for this association of complement defect and an autoimmune immune complex disease is unknown, but it may be related to the inadequate clearance of circulating immune complexes because of defect on complement activations. If normally generated immune complexes are not cleared from circulation, they may be deposited in the blood vessels, wall, and tissue, where they activate the leukocyte by Fc receptor dependent pathway and produce local inflammations. Complement may also play an important role in the clearance of apoptotic bodies containing fragments of DNA. These apoptotic bodies are likely source of the nuclear antigen that trigger autoantibody response in lupus. In addition, complement proteins regulate antigen-mediated signal received by B cells. In their absence, self-antigen may not induce B cell tolerance and autoimmune results. Some patients with C2 or C4 deficiency show increased susceptibility to infection, and other are asymptomatic. Deficiency of C3 is associated with frequent serious pathogenic bacteria infection that may be fatal, illustrating the central role of C3 in opsonization and enhanced phagocytosis and destruction of these organisms. Deficiency in component of alternative pathway result in increased susceptibility to meningococcal infection. Factor B and factor D deficiencies are rare, but X-linked recessive propedin deficiency is more common. Genetic variant of gene encoding MBL and MASP contribute to the immunodeficient in some patients. This is discussed in chapter 21. Deficiency in terminal complement component, including C5, 6, 7, 8, 9, have also been described. Interestingly, the only consistent clinical problem in this patient is propensity of disseminated infection by Neisseria bacteria, including Neisseria meningitis, Neisseria gonorrhea. As mentioned earlier, complement-mediated bacteria is particularly important for defense against these thin-walled organisms. Deficiencies in complement regulatory proteins are associated with abnormal complement activation and a variety of related clinical abnormalities. Deficiency in C1 inhibitor DAF causes hereditary angioedema and paroxysmal nocturnal hemoglobinuria, respectively, mentioned earlier. In patients with factor 1 deficiency, plasma T3 is depleted as a result of unregulated formation of fluid phase 3 convertase by normal takeover mechanism. The clinical consequences increased infection with pathogenic bacteria. Factor H deficiency is rare and is characterized by excessive alternative pathway activation, and C3 and glomerulonephritis caused by inadequate clearance of immune complexes of renal deposition of complement byproduct. A form of hemolytic uremic syndrome involved defective complement regulations, and most common mutation in this condition are factor H genes. Another genes that is mutated is in some patients is the Mcp genes. In this disease, children present with microangiopathic hemolytic anemia, thrombocytopenia, and acute renal failure, all triggered by endothelial cell injury caused by hyperactivation of an alternative pathway of complement. Mutant factor H and MC bind less well to C3b and C4b on endothelial surface, and as a result there is excessive complement activation leading to the formation of microthrombi and vascular damage. Mutation in factor I and factor H result in unregulated activation of alternative complement pathway, kidney disease collective called C3 glomerulonephritis. This is similar to the effect of autoantibody called C3 nephrotic factor, C3-NEF, which are specific for alternative pathway, C3 convertase, and C3-NEF stabilizer, T3BB, and protect the complement form of factor H mediated dissociation, which result in unregulated activation C3. Specific allelic variant of factor H are associated with age-related macular degeneration. Excessive inflammation in the absence of complement regulation contribute to the disruption of photoreceptor cell in the macular region and consequent blindness. Mutation in PIG-A phosphatidylinositol glycosyltransferase A genes result in paroxysmal nocturnal hemoglobinuria, as discussed earlier, as a result of defective GP anchor for CD59 and DAF. Deficient in the complementary receptor include absence of CR3, CR4, both resulting in the rare mutation in beta chains CD18 that is shared by CD11, CD18 family of integrin molecule. The disease caused by this gene defect is called leukocytization deficiency, seen chapter 21. This disorder is characterized by recurrent pathogenic infection and is caused by inadequate adherence of neutrophil to endothelial tissue sites and infection, and perhaps the impaired IC3b dependent phagocytosis of bacteria. Pathological effect of immune complement system. Even if it is properly regulated and appropriately activated, the complement system can cause significant tissue damage. Some of the pathological effects associated with the bacterial infection may be due to complement-mediated acute inflammation response due to infectious agents. In some situations, complement activation is associated with intravascular thrombosis and can lead to the ischemic injury to tissue. For instance, anti-endothelial antibodies against vascularized organ transplant, and the immune complex produced in autoimmune disease may bind to vesicular endothelium and activate complement, thereby leading to inflammation and generation of mark with damage of endothelial surface, which favors coagulations. There is also evidence that some of the late complement proteins may activate prothrombinases in circulation and initiate thrombosis independent of MAC-mediated damage to endothelium. The clearest example of complement-mediated pathological condition are immune complex-mediated diseases. Systemic vasculitis, immune complex glomerulonephritis result from deposition of antigen-antibody complex in the wall of blood vessels, kidney, glomerulus. The complement activated by these deposited immune complexes initiate an acute inflammatory response that destroy the vessel walls and or glomerulus, lead to the thrombosis, ischemic damage to tissue, and scarring. Studies with the knockout mice lacking the complementary protein C3, C4 or lacking of Fc gamma receptor suggest that Fc receptor mediated leukocyte activation may also cause inflammation and tissue injury as a result of IgG deposition even in absence of complement activation. The two therapeutic agents that target the complementary system are currently approved by clinical use. Antibodies against human C5 that block the proteolytic cleavage of C5 are used to treat paroxysmal nocturnal hemoglobinuria, complement-mediated hemolytic uremia syndrome, and neuromyelitis optica, and are also being investigated as therapeutic in number of chronic inflammatory disorder. Recombinant human CH is used to treat patients with hereditary angioedema. Evasion of complement microbes. Pathogens have evolved diverse mechanisms for evading the complement system. Some microbes expresses thick wall that prevent binding of complementary protein, such as MAC. Gram-positive bacteria and some fungi are examples of microbes that use these relatively non-specific evasion strategies. A few of more specific mechanisms used by selecting pathogen will be considered here. These evasion mechanisms may be divided in three groups. Microbes can evade the complement system by recruiting host complement regulatory proteins. Many pathogens, in contrast to the non-pathogenic microbes, expresses celiac acid, which can inhibit the alternative pathway of complement by recruiting factor H, which displaces the C3b from BB. Some pathogens like schistosomes, N. gonorrhea, and certain Haemophilus species scavenge celiac acid from the host and enzymically transfer the sugar to their cell surface. Others, including Escherichia coli, K, and some meningococcal, have evolved special biosynthesis routes for Fossilic acid generation. Some microbes synthesize protein that can recruit regulatory protein factor H to the cell surface, GP41 on the human immunodeficiency viruses, and can bind factor H, and this property of virus is believed to contribute to the virion's protection. Many other pathogens have evolved proteins that facilitate recruitment of factor H to their cells walls. These include bacteria such as Streptococcus pyogenes, Bacteria butogopheri, the causative agent of Lyme disease, N. gonorrhea, N. meningitis, fungal pathogens Candida albicans, and nematodes such as Echinococcus granulosus. Other microbes such as HIV incorporate multiple host regulatory protein into their envelope. For instance, HIV incorporates GPI, anchoring complement regulatory proteins DAF and CD59 when it buds from uninfected cells. A number of pathogens produce proteins that inhibit different steps in complement activation. E. coli makes C1q binding protein that inhibit the association of C1q, C1R, C1s. Staphylococcus aureus takes a protein called Staphylococcus complement inhibitor that bind to and stably inhibit both classical and alternative pathway 3 convertase and thus inhibit 3 complement pathway. Glycoprotein C1 of herpes simplex viruses destabilize the alternative pathway convertase by preventing its 3b component from binding to propedin. GP160, a membrane protein trypanosoma clones, the causative agent of Chagas disease, bind to C3b and prevent the formation of T3 convertase and also accelerates its decay. Vaccinia viruses complement inhibitory proteins structurally resemble human C4BP but can bind to both C4b and C3b and accelerate decay both T3, C3 and C5 convertase. Complement-mediated inflammation also can be inhibited by microbial product. Staphylococcus aureus synthesize a protein called chemokines inhibitory protein staphylococcus chiefs, which is an antagonist of C3a anaphylactosin. Summary: humoral immunity is mediated by antibodies and is the effector arm of adaptive immune system responsible for defensive against extracellular microbes and microbial toxins. The antibody that provide protection against infection may be produced by long-lived plasma cells generated by fast exposure to microbial antigen or reactivation of memory B cells by antigen. Antibodies blocks or neutralize the effectivity of microbes by binding to microbes and is theoretically hindering the interaction of microbes with the cellular receptor. Antibodies similarly block the pathogenic actions of a toxin by preventing binding to the toxin to the host cell. Antibody-coated opsonized particles are phagocytized by binding of the Fc portion of antibodies to phagocytic Fc receptor. There are several types of Fc receptors specific for different subclasses of IgG and IgA and IgE antibodies, and different Fc receptor bind the antibody with varying affinities. Attachment of antigen complex IgG to phagocyte Fc receptor also deliver signal that stimulate the microbial activities of phagocytes. The complement system consists of serum and membrane protein that interact in a highly regulated manner, produce biological active product. The three major pathways of the complement activation are alternative pathway, which is activated on microbial surface in the absence of antibody, the classical pathway, which is activated by antigen antibody complex, and lectin pathway, which is initiated by circulating lectin binding to a carbohydrate on pathogens. These pathways generate enzymes that cleave C3 proteins. The cleaved products of C3 become covalently attached to microbial surface or antibodies. So subsequent steps of complement activation are limited to these sites. All pathways converge on the common set of late steps, which involve the proteolytic cleavage of C5 and culminate in the formation of membrane prawns. Complement activation is regulated by various plasma and cell membrane proteins that inhibit different steps in cascade. The biological function of complement system include opsonization of organism and immune complexes by proteolytic fragment of C3, following by binding to phagocytes receptor for complement fragment and phagocytic clearance. Activation of inflammatory cell by proteolytic fragments of complement protein called anaphylactosin T3, C3A, C4A, C5A. Cytotoxic mediated by membrane attack complex formation on cell surface, solubilization and clearance of immune complex, and enhancement of humoral immune response. So you will find more further readings here below. They are good studies that you can pause and enjoy. The next topic will be spoken later. There will be many more.